Okay, so how I work these is that we're going to go through and you guys can ask me specific questions and then I'm going to, talk about them. We can do anything and go back and do it again if you want to at the end, but I'd like to try to go through them at least at the beginning here by chapter. So we're going to do all of our chapter 5 stuff first, then the chapter 7 stuff, then the chapter 8 stuff, and if there's time, whatever. Then I can go back and ask some more questions. I just don't want to be jumping back and forth between all the topics. This is when you start asking questions. Okay, so what specifically so what do you know and what's the and where is it breaking down? So, a complementation test is for alleles that have the same mutant phenotype. So they have to have the same mutant phenotype and they have to be recessive, because if they're dominant, then all bets are off. The end goal of a complementation test is always to put the two alleles that you're asking whether they're alleles of the same gene or not in trans to one another. So in whatever set of crosses you have to do that, usually like this, they'll just be crossing one to another, unless you're working with like sterile genes like we're doing in the lab. But these are fine, well, we don't even know what they are. But you take one mutant homozygote, that's A, it's homozygous for little a, it's going to have the mutant phenotype, cross it to another individual that is homozygous for mutant B, it's got the mutant phenotype too, then because this individual on the left and the individual on the right are both homozygous, then they can each only give one allele, right? So there's only one genotype possible for the progeny. It's a one square Punnett square. So the progeny all get a chromosome with the A mutant, little a mutant from the left parent, and little b mutant from the right parent. And then the question is the mutant phenotype seen or not? If each of the two alleles being tested are both alleles of the same gene, then that gene is broken in both alleles. So here, the A mutation in the top chromosome can't make a good gene product, and the B mutation in the bottom chromosome here also can't make a good gene product. And so essentially you are homozygous for mutations in this gene, just like this is homozygous for a mutation in that gene, and this one is homozygous for a mutation in that gene. It's just that here you're homozygous with the same mutant allele, and same thing here, but here you would be homozygous mutant for that gene. It's just that the top chromosome and the bottom chromosome have different mutant alleles, but they're mutant alleles of the same gene. You have no good copies of that gene, therefore there's a failure to complement, right? This example is if the alleles are with different genes. In the same way as the previous slide, this progeny down here is the product of a 1 square Punnett square, because the left parent is homozygous and the right parent is homozygous here. And so what you've got down here is you're putting, again, mutant allele A in trans to mutant allele B. However, in this case, A and B are not alleles of the same gene. And so you're not homozygous mutant for any gene in this progeny down here. In the left parent, you're homozygous mutant for A, and in the right one, you're homozygous mutant for B. But because each of these are only mutant for one gene, the left one is wild type for B and the right one is wild type for A. So when you're down here, these chromosomes complement each other. They make up for each other's deficits. The top chromosome has a mutant A, but the bottom chromosome has a wild type A, and that means that you're heterozygous for the A gene. You're not homozygous, and so you wouldn't show the mutant phenotype there. Over here, the bottom chromosome is mutant for B, but the top chromosome complements it because it's wild type for B. So again, you are a heterozygote, and so you are heterozygous for both A and B, and you show the wild type phenotype there. You're nodding. Is that real? Okay. So when I want you, so this is tricky because it's just like what we talked about in lab last week, in that there's two possible outcomes when you do a complementation test. I'm telling you what happens if the two alleles are the same, or from the same gene, and what happens if they're from different genes. But the purpose of a complementation test is to answer that question. You would never do a complementation test if, like in both of these examples, you already knew the answer. You're doing the complementation test to find out, it's just my cup. You're doing the complementation test to find out the answer. And so you're doing the cross and seeing what happens, but you know what would happen if they are the same or different genes. And you just see which one that matches up with. So you always, in these cases, for a complementation test, you always want to take two homozygous mutants, cross them to each other, get the trans heterozygote, or put the mutant alleles in trans to each other. In this case, it's not, back up there. In this case, it's not a trans heterozygote because it's the same gene. So in both of these cases, you're putting the A allele and the B allele in trans to one another. It's just in one case they're both alleles of the same gene, and in the second case they're not. So then the answer for all these and that we did for the practice problem is that in order to see if any new gene is an allele of either W1 or W2, you would just cross homozygous mutant A into mutant W1, not the W1 plus, right? And you would cross B into the mutant W1, and you would cross C into the mutant W1. That would be the complementation test to find out if any of these were mutant 1 or were W1. Then you would have to do A, B, and C with mutant or with W2. Unless any of them showed failure to complement with W1. Because if you show failure to complement, let's say there's 10 genes on this chromosome that if you mutate get white flowers. Once you fail to complement one, the game is over. The only thing you would want to do is redo the test with a positive control maybe to make sure you did the cross correctly. Because if your mutant fails to complement one tester, if your new mutant fails to complement, did everybody sign this? If your new mutant fails to complement one of the testers, then that's it. You know it's an allele of that gene, and therefore it can't be an allele of anything else because, right, alleles are only an allele of one gene. Okay. All right, let me go to where it's back up here. Okay.
So my question is, I know this might sound crazy, but how would you know if it's a recessive or if it's going to be a dominant allele that's going to be allele?
Well, I will tell you. In this case, I will tell you whether something is dominant or recessive, for if it's going to be something like this. However, if you do a cross here, you would know that this is dominant because you cross two yellows and you get two yellow and one non-yellow in the progeny. That can't happen if they're recessive, right? And also, the other thing that was known about this case is that you can get, so you get two yellows and one non-yellow in the progeny. The non-yellow or the white or the gray or whatever that is down there is true breeding. But it's impossible to get a yellow true breeding even though you have yellows going. So that means you're never homozygous for big Y. All these guys up here have to be heterozygotes because you, whoops, back up there, because to get a non-yellow you're going to have to be homozygous. And so these two both have to have the little y allele. It can't be one of them's big Y, and one of them's big Y, little y, because if that was the case, everybody'd be yellow, and the game would be over. Let's say that it was, well, see, that would be tricky. If little y was lethal, you would never be able to see it. In a cross like this, you would mistakenly think that you had homozygous big Y, big Y. I mean, everybody would be yellow all the time. So you'd be big Y blank and you'd be yellow, but you would never see any mice that are not yellow because they would all fail to develop. So hold on. Yeah, all right. We're save it for, we're going by chapter. So what else in chapter 5? Now, that doesn't mean like when we go through all of them, if you remember something, we can go back at the end, but I just want to go through in the correct order the first time. Yep. So the difference between, duplicate recessive epistasis and duplicate interaction, right? Which is going to be this one here. Well, I will go back and say, for anything that's epistasis, you know that it's epistasis and not a lethal allele because you still get the right number, the right total number of classes, right? You get a 9 to 3 to 3 to 1. You get a total of 16. Then to figure out what's going on, you have to look at what gets combined. So in each case where there's epistasis, you get two or more of the classes combined together. In the case with duplicate recessive, you see 9 and 7. That means that the 3, and the 1 are all together. So just thinking about that without knowing anything else, you can figure out what is epistatic and what is hypostatic. Because the 9 has at least one big A and at least one big B, there are no little a homozygotes and no little b homozygotes in the class that has the brown color. But in the albino snails that lack color, you always have at least one little a homozygote or one little b homozygote, or in the case of the one on the far right there, both. And so you can tell then that being recessive for little a, or being homozygous recessive for little a, is epistatic to B. So no matter what you have for the B gene, you're going to be no color if you're homozygous recessive for little a. And little b does the same thing. So in this one here, little b is epistatic over big A blank. So it's little a can make whatever happens with B unimportant or not count because it's masking it, and little b makes what happens with the A gene unimportant because it's masking it. In order to see any color, you have to have at least one big A and one big B allele. So 9 to 7, because the 3, and the 1 are together. Now, in duplicate interaction, what happens is you have two extreme classes, either the completely flattened out or the completely long crookneck squash, or these little spherical 8-ball guys in the middle, right? And here, if you look down where it is right there, so your phenotype ratio becomes 9 to 6 to 1. And so instead of 9 to 7, so if you can see that if you have one big A and one big B, as the first one does, then you're going to be the completely flattened out shape. And if you have none, you're the completely long crook neck. And then you're like, okay, so these are in the middle, but it's weird because one of them has big A blank and little b's, the other one has big B blank and little a's, but yet they both give the same phenotype. So you're having an interaction between these genes. And what I said in class was that the answer is that each big allele gives a certain amount of flattening power, so to speak. However, whether you're heterozygous or homozygous for A, big A, or whether you're heterozygous or homozygous for big B doesn't matter. All that matters is if you have one. If it was homozygous mattered, there'd be a bunch of more, there'd be more up here, there'd be more classes instead of just the very squished out, the middle, and then the very long. Yeah, question. Is duplicate interaction kind of sort of what? Duplicate interaction is like incomplete dominance phenotypically. It would be incomplete dominance if there was only one gene involved. So in that case it would be if you're big A, you're the flat. If you're big A, you're the disc shaped here. You're little a, you're this. And if you're big A, you're that. How can you tell that that is not duplicate interaction? You look at the numbers. So duplicate interaction is a 9 to 6 to 1 phenotype ratio. Big, medium, and small number, right? If you cross two heterozygotes, like if you took a big A, little a here, and it was the situation I explained a second ago, what phenotype ratio of flat, sphere, and long would you get then? One to 2 to 1. You don't get 3/4 because there's an intermediate, right? When do you get 3/4? When there's complete dominance. So if you have complete dominance, all right, let's look. If let's say big A is the disc shape and little a is the long shape here. If you had complete dominance and crossed a big A little a to a big A little a, then 3 of them would be big A blank and be the disc shape. 1 of them would be little a and be long, and this wouldn't exist because you have complete dominance. In complete dominance, having one dominant allele is just as good as having 2. This is where, at the beginning we had a little chat in class about, are the exams cumulative? And I said, well, I say no, but really they are. And we talked about it in the same way that a math class is. So we're having to remember what straight up complete dominance is. Now we got incomplete dominance. Now you've got duplicate interaction, and you're looking at these ratios. 1 to 2 to 1 is a lot different than 9 to 6 to 1. Right? It's a small, a medium in the, small, bigger, and small, right? Versus big, a little bit smaller, and really tiny here. So when you get when you get anything that adds up to 16, then there's going to be 2 genes involved. If there's only 1 gene involved, and you cross 2 heterozygotes to each other, there's only 2 possibilities that can result. 3 to 1 phenotype ratio or 1 to 2 to 1 phenotype ratio. Because no matter which phenotype ratio you get, you're always going to have a 1 to 2 to 1 genotype ratio. Okay? This is the thing. So it's like, don't try to memorize this. Because from what I just went through with you, you can, if you know what you're doing, get some confidence from working these problems and really understanding what's going on. You could figure anything out just by looking at the numbers. So what's the only thing we've talked about that could give that kind of a ratio? It's duplicate interaction, right? And with any of these epistasis examples, any of them, you can look at the classes that are combined and know what genotypes are supposed to go in those classes, and then figure out what is going on with the genes to give the phenotypes. Come on up here and sign this. And then, I don't have a pen, so bring something. Oh, actually I do. Never mind. Sorry. So the epistasis, and I said, oh, look at this big bad table, right? So don't try to just memorize this. Learn what happens in each of the cases. And then when I give you something on the exam, you can figure it out. So what goes to, what are the what phenotypes go with which possible genotypes, and how could that work itself out? And remember, epistasis is one gene masking another one. It's not dominant and not recessive, right? Because you can have dominant epistasis and recessive epistasis. I already did that, and I'm recording this, so you've got an expert note taker right here who was paying attention, and it's also recorded. So come on, say it again. Yes. Everything we talk about in class is you have to know about. So yes, you do have to know about cytoplasmic inheritance. And it's just that each of the what? Well, where is your understanding breaking down? Don't look around. You asked. So it does. So what do you know? See, this is the thing. What do you know about it, and where's your understanding breaking down? Because, you have to try it and figure out where things aren't working before I can help you on it, because I already explained it once in class, and if I go over it again, it's going to be the same thing. But that's why I'm asking you. So yeah, yeah. Yep. So how does the replication of mitochondria and chloroplasts work? And how is that different? How is the replication of and inheritance of mitochondria and chloroplasts as organelles different from chromosomes? How are the chromosomes partitioned during meiosis or mitosis? Right, they line up, and then what happens? They separate. How do they move? They're not just floating around, are they? Well, how do they move then? In either case. Okay, yeah. I'm not talking about which. So the chromosomes or the chromatids move to opposite poles of the cell. The opposite poles of the cell do not just have a magnet and is attracting them. How do they move? The what? Right, the spindle fibers, the microtubules, hook onto them and pull them apart, right? And half of either the homologous chromosomes or the sister chromatids end up in each half because they're attached, they line up at the metaphase plate, and they get pulled to the other side. Chloroplasts and mitochondria are, I don't want to say free-floating because that's not completely accurate, because the cytoplasm is really more of a gel than like a glass of water. But you can think of it that way. As the cell grows in size to be big enough that it can split into two, the mitochondria, if you're an animal, or the mitochondria and the chloroplasts, if you're a plant, are both duplicating and replicating. And they get separated approximately half into each daughter cell just by chance. Half the cytoplasm goes to one daughter cell, the other half the cytoplasm goes to the other daughter cell. When this pinches off here, It's just the half of them that are over on the left go to that cell, and the half that are over on the right go to that cell. So if you get a mutation, like the red one, let's say, is a mutation, it depends on which ones get replicated more than the other ones, and the random moving around of the organelle before the cells undergo cell division to determine which ones get into which daughter cell. And so the cell that ends up with all red ones at the very bottom right down there, we were calling that in class, that's the one with the mutation, and they're all going to be mutant there, is just random and accidental. It just so happened that that one got all the red ones. Well, actually, here, this one got all the red ones and that one got all the blue ones, and now this lineage stays blue, and this lineage stays red, because they're, actually, here the cell's dividing, and down here the mitochondria are replicating. But here they're all blue because the red is a mutation and it hasn't had another mutation yet. This one will stay all bad unless a back mutation happens where you get lucky and those mitochondria, get a mutation that puts it back the way they used to be. But if this is a severe enough mutation, that cell with all the red ones, because they're mitochondria, is going to be energy deprived, and it's going to go down in, it's not going to be very healthy, to the point that it will be recognized as defective by the cell, and hopefully the apoptosis pathway will be activated and it'll get killed. Now, in some examples where you inherit all your mitochondria from mom, and there are some mutations that are not lethal but still leave you in very bad, in a very bad shape. And so in those cases, the reason why you as an individual would have all defective mitochondria in all of your cells is because this you could think of as like mitosis, but this can also be the same thing during meiosis. And if the final egg has, all defective mitochondria, then you're not, that's what you're going to get. Because you start out with one oogonium and it keeps all the cytoplasm during oogenesis, this cell division would have to happen sometime during the mitotic phase, because you're not separating cytoplasm during oogenesis, remember? You're going from one big cell that's 2N down to one big cell that's just N, and you get those two polar bodies that are like little bumps on the outside of the mature egg, and they contain nothing but chromosomes that are just going to get disintegrated and get thrown away. And when you think of this, of course, if you have all good or all good chloroplasts, all your daughters will. If you have all defective, all your daughters will too. Now look, remember this one has green leaves down below, but has white leaves here, and so at each axis, I can't remember what they call it, where you get a node, at each node where you get a splitting there in the plant, this is all derived from the same cell. And so if these leaves are all white, that means all the reproductive parts up here are going to be white too. And if you could have a plant that was sort of a little bit of both, like see this one has a white leaf down here, but up here they're variegated, that means some of the cells have all defective chloroplasts and are the white patches, and some cells probably have a mix of good and defective chloroplasts in their green. Now some of those are going to end up having all good. Where's my pointer here? It keeps disappearing, because you can get all white, all green, or I would say this is the most likely coming out of that. And it just depends on the random, pretend these are chloroplasts now and not the mitochondria appearing. It just depends on the random segregation of chloroplasts in the mitotic phase of gametogenesis. Okay, now we're in Chapter 7. So what are your questions about Chapter 7? Linkage and gene mapping by means of recombination frequency. How do I determine what? Oh, sure. I'm sorry, guys. I mean, I'll ask you, just speak up because I got one ear and that is louder than... So let's start here. So which locus is going to be in the middle? If you have a double crossover, if you look at just panel C there, if you have a double crossover that involves one crossover between A and B and a second between B and C, you can see that only B is changed. And if you follow with your finger here, little a, big B, little c, only big B is different. Big A, little b, big C, only B is different. So the gene that is different is the one that's going to be in the middle. Now you have to come down here and using this progeny table, decide which are the non-recombinant or parental type. That's the ones with the most, because if genes are linked, they're going to stay together more than they're going to separate apart. And then which ones are the double crossovers? That's the class that has the least. Because if you have a certain probability of recombination between A and B and a certain probability between B and C as single crossover in recombinations, then in order to get one between A and B and C, that's going to be more rare because you have to get one between A and B and you have to get a second one between B and C occurring at the same time. So the biggest class is going to be those like this chromatid here and this one down here that are not involved in recombination. Then there's going to be two intermediate classes. In that case, it's this one and this one. One of those is going to be a recombination event between the left and middle. The other one's going to be a recombination event between the middle and the right. Then there's the smallest one, the one that's circled with the 5 and the 3. That's going to be the rarest, and the reason we know that is because it's got the least. So then we look at the chromosomes for the one that's got the least, and you see here's shoot, what is the, I don't remember what ST is. Oh, red eyes and scarlet. It's scarlet. So scarlet and ebony are the same here, and spineless is different. Then go to the other one and check your work. Here, Scarlet and ebony are the same, and spineless is different again. Therefore, you know that spineless is the one that's in the middle. You've got to figure out which class has the double recombinants. In other words, which one's the smallest before you look at anything else. Because if you look at this one, look, here's two that are the same, and now scarlet's different. Check yourself. Here's two that are the same, and scarlet's that's different, right? Every one of them that's not the non-recombinant parentals is going to have two that are the same and one that is different. And so if you pick the wrong class, you're going to get an answer, but it's going to be wrong. And that's how you figure out which one's in the middle. Yep. So there's going to be one that's two that are the same and one that's different, right? So in this case, you're going to compare to the wild type parent because there's two of them here, right? You compare it to the parent that matches the two. And you don't even need to look at the parent, right? Because look, here's two that's the same that are wild type and one that's different, a mutant. On this one, here's two that are the same, mutant, and one that's different, wild type. Now you could take this one and compare it to this, the all mutant, and you can take this one and compare it to the all wild type, but you can tell just by looking at this which one's the one that's different. The important thing is choosing the correct class, because if you choose incorrectly, then you're not, it's not going to work out. So, in problems you get from me, you'll be able to tell. Now, so let's put these in the correct order here. In this case, between scarlet and spineless is 14.6 and spineless to ebony is 12.2. But what if they were exactly the same distance, right? Then you'd be stuck. You'd still be able to tell the wild type from the smallest class, but these two classes would be so close that you might not be able to tell one from the other. That's never going to be the case in a problem that I'm giving you. If it would ever be the case in an experimental situation, you just wouldn't be able to figure it out. I mean, I guess you would from having large enough numbers and doing doing a chi-square test to make sure that you pick the right one for the right. But if they were exactly the same distance and there was no recombination hotspot issues and they gave exactly the same recombination frequencies, then no, you wouldn't be able to tell which is which. And what you would have to do is do some more crosses, exchange out either one of the mutants for something else that would give a different number. If they're exactly the same, then no, you wouldn't be able to tell. You wouldn't be able to tell which was which. But honestly, then it wouldn't matter in this case anyway, because you don't know whether this is right or left, right? If you do the whole chromosome, you would have to figure out something else over here and something else over here to tell which orientation this is in. If scarlet is really on the left and ebony is on the right, or if ebony is on the left and scarlet is on the right. It's going to be obvious, so I won't put them together like this and like this. But even if you had these, in any kind of order you put them in, you'd be able to tell that 50 and 52 go together and 41 and 43 go together, and it wouldn't be 50 and 43 and 41 and 52. It doesn't make sense. They will be obvious enough that there shouldn't be any question. this? Okay, so what is your question? I'm sorry, could you speak up a little bit? Just come up and ask me and I'll repeat it. And I'll repeat it for the recording.
For this one, I was just not understanding how this became this.
Oh, okay, all right. So the question is, how do these two parents produce all of these gametes down here? So when you're doing a test cross for gene mapping, you're always going to be crossing a heterozygous individual like this one to a homozygous tester plant. The homozygous tester plant, because it's homozygous recessive for both genes, is only capable of producing one gamete type. It doesn't matter. Yes, recombination could happen between the M and the D loci, but if it does, you would still have little m and little on the left and little d on the right. There's nothing else there. So this one is just there to provide a chromosome with the two recessives. Now here, these two are linked on the same chromosome. So more often than not, you will have this chromosome going to one gamete and this one to another, as is shown here. And I guess in here. But that's, if there's really no recombination, you get this, right? Now in some smaller fraction of cases, you get recombination. So let's go from left to right here, big M, and then a recombination event here to little d. And little m up here to capital D, right? And so that is big M and little d, little m and capital and big D right here, right? The reason why you get some of these non-recombinants is, let me find the picture. Because even when recombination occurs, 50% of the progeny are non-recombinant. I'm sorry, what? No, so recombination happens before anaphase of meiosis 1. There is no recombination in meiosis 2. So here, look. When you're talking about recombination, remember you're talking about replicated or duplicated chromosomes. There's two chromatids on each one. This one's got big A, big B here. This one's got little a, little b. But if you get a crossing over event here, it's only between two of the four chromatids. So you're going to get big A, little b, right? And little a, big B. So this is big A, little b. There's that one. Little a, big B, that's two. But this one up here and this one down here did not undergo recombination. So big A, big B right there. Little a, little b down there. So in an event where you get a single recombination, 50% of the gametes are recombinant and 50% are not. The 50% that are not recombinant because they are the chromatids on the homologous chromosomes that did not participate in recombination. That was your question, right? So let me go back to this really quick so I can just make sure that, so then if there is no recombination, then half the gametes are this and half are that right here. If there is recombination, then in whatever fraction you get, recombination for, half of the gametes will still be non-recombinant and half will still be recombinant. But this fork versus this fork, they're different percentages. You're always going to get more of these, the non-crossing over ones. So what was your additional question? What figure are you talking about? Like this? This?
Yes, I think it is that. This one.
So, if you have recombination, 50% of the gametes are recombinant and 50% are not. We just talked about that. I think what you're confused about is, so then how do you tell that from independent assortment, right? The way you tell from independent assortment is, most of the time, this doesn't happen at all between A and B. So far more times than this happens, this little recombination event here won't occur, and 100% will be parental and 0% will be recombinant, right? That's what I'm talking about. So here, 50% are recombinant and 50% are non-recombinant, only of those where a recombination event actually takes place between the two genes. In the majority of the cases, a recombination event will not take place between the two genes, and therefore you would get 0%, which is, if this didn't happen, you'd get two gametes that look like this one, big A, big B, and two gametes that look like this one, little a and little b. So, either I can give you numbers of progeny, you add them up, and then divide the recombinants by the total, or I will tell you the recombinant frequency and the total number of progeny, and you just multiply the progeny by the recombination frequency to get the recombinants, and the frequency of non-recombinants is always 1 minus the frequency of recombinants. It's just 1, right? You can't have more than 100%. So if the recombination of frequency is 10%, then that means 90% do not have recombination. And if I said I gave you we did a cross with a heterozygote of two genes that are 10 map units apart to a homozygous recessive tester, and then picked 300 progeny, how many of them should be recombinant? And it would be like 30. out of 300 because it's 10% recombinant. So 270 would be non-recombinant and 30 would be recombinant. So there's a backwards and a forwards way to do it. Either I give you the final numbers and you calculate the recombination frequency, or I give you the recombination frequency and the final total, and you tell me how many go into each category. The place where you got to be careful is, oh, it's after this. Here. This is a 16% recombination frequency, but you've got to keep in mind that each of the two recombinant classes is only going to be half the recombination frequency. And then each of the non-recombinant classes is going to be half the non-recombinant frequency or parental frequency. Save it for the end. We're going through the chapter by chapter. So we've already done chapter 5, but nobody's asked about that yet, so we'll bounce back to it at the end. Ask again then. So any more for chapter 7 now, then we'll go through chapter 8, and then it'll be like a free-for-all for anything that got missed. I just don't want to bounce back and forth between chapters. So anybody else for chapter 7? Are we moving on to chapter 8 then? All right. So any questions about the parts of chapter 8 that we've talked about so far? So we've talked about the chromosomal rearrangements, which just affect one chromosome, unless you're talking about a translocation, which can affect two. We've sort of halfway gone through aneuploidy, and then we're going to talk about polyploidy in the end, like we talk tomorrow. So, questions about what we've gone over so far? Yep. Okay. So you said pericentric with an I, right? So in a pericentric recombination, the centromere is inside where the break and inversion occurred. Just like with the pericentric, when you're done, if you get a single recombination event within the inversion, you can be in trouble because the inversion loop formed. It's this twist here in the inversion loop that causes all the trouble. So instead of the pericentric, where you get an acentric chromosome that's lost, and then the trouble for all the gametes is that some genes are lost, here you're going to have an exchange between the non-sister chromatids, but it's going to be an unequal exchange. One chromosome is going to give up some genes to the other one, but it's going to get back other genes that aren't the ones that it gave up. So it's a crossover and recombination event in which non-identical parts of the chromosomes are exchanged. Then you get a lineup that looks like this, because this one here that I'm highlighting, let me make it bigger. This one that I'm highlighting gave up its purple and gray and exchanged it for red and orange, and that's why it's bent around like this. It should be flopped down here and have purple and gray and be lined up against the other one there. This one that I'm highlighting now, moving the mouse over, is the one that gave up the orange and yellow, or I'm sorry, red and orange, and got a purple one here and a gray one there instead. So it's got two purples and grays and no red and yellow. This one's got two red and, well, orange and no purple or gray. This is just regular synapsing right here and here. Just like what would happen if these chromosomes were lined up normally and no inversion had taken place. So when the sister chromatids pull apart and this, or when the homologous chromosomes pull apart and this centromere goes up, and this one goes down, then you get a peeling off here and a peeling off there, and you end up with these these chromosomes that have unequal parts on them. See, this one's got a red and an orange here, but there's no second version of it. It's got the gray and purple instead. And same thing with this one. So now when you go to make the gametes, these two in the middle are non-viable. So just like the paracentric, you've got 50% viable and 50% non-viable, but here these are non-viable because they have two of some genes and none of others. So when they get to be part of a zygote with a normal gene, the gray and the purple here, there will be 3 copies of that instead of 2. That's a problem, gene dosage. And you also get a gene dosage problem with the red and the orange because instead of 2, there's only 1 now. Make sense? And same thing with this one, come on pointer, except it's going to have 2 copies of the red and orange and none of the purple and gray. And these two, the top one that doesn't have the inversion and the bottom one that does have the inversion, are just fine. The reason why they're just fine is because both of them have 1 copy of every gene that is on that chromosome. And then the reason why they have 1 of everything is even though they were involved in the inversion loop, no recombination took place. So if you get recombination taking place over here, or a double recombination in the middle, nothing's going to happen. You're going to be just fine. Any swapping of these parts is going to be equal, the red and the orange. Any swapping of the purple and gray parts over here is going to be equal. Everybody will be fine. If you have a single crossover here, there's a problem. But if there's another double crossover between the same 2 sister chromatids, then it puts everything back and you're all right. There will be a little bit of exchanged information here, but the exchanged information will be the same genes, and so you're just fine. The what? Oh, okay. So what happens, so when you have an inversion heterozygote like I've got shown here, if you get a recombination within the inversion, those gametes that were involved in the recombination are dead. They're not viable, right? So that allows you to hold something over the inverted part here that you don't want recombination to happen for. So here, we don't want recombination to happen between UNC54 and FIR14. If we just had a wild type chromosome down here with pluses, you could get a recombination that occurred that would allow you to have paralyzed worms that were not sterile or fertile, right? We don't want that. So by having and this chromosome up here has no inversion, just the UNC54 and FIR14. The purple that I've got here is just showing where the inversion would be, like what we call covered by the inversion. But any recombination between this top chromosome and the bottom one within that purple region, those recombinants will be dead. That allows us to keep UNC54 and FIR14 together. They're both recessive, so that anytime I see a paralyzed worm on the plate, I can just pick the paralyzed worms, which you can see, and know that it will be sterile, which you cannot see until you check them by letting them have babies and seeing that they do not. The thing that I've added down here is that the inversion chromosome has another allele that is lethal. Yes, it is a UNC gene, so some alleles of UNC75 are paralyzed because they just have point mutations in them, and the protein can still work a little bit. But if you have a mutation that completely kills the protein function, it's dead. So this would be like a haploinsufficiency thing, except that this isn't dominant, right? And so that means that the inverted chromosome is dead as well. If you just had an inversion here with no mutations in it, then this would give paralyzed sterile ones, heterozygotes that look wild type but would give you more paralyzed sterile ones, but it would have inversion homozygotes that would also look wild type. So we have some of those in the old time strains before people did cool stuff like this, and even cooler stuff by putting a gene for a green fluorescent protein in here, so everybody who's a heterozygote glows green, and then everybody who's not is going to be sterile. So this guy is dead as a homozygote. So you know that everybody who is not paralyzed has to be a heterozygote, because homozygotes for this chromosome are paralyzed, and homozygotes for this chromosome do not exist because they're dead. Got it? So the purpose of a balancer is to be able to maintain an allele like this one that is sterile, or an allele that is lethal. And why would we want to maintain a lethal allele? Because lethal alleles are everything up to and including, some people include sterile mutants as lethal as well. Lethal is anything that doesn't go to the next generation. Most lethals die sometime while the worm is an egg. But you can tell where the gene product functions by seeing how far they go. The complete lineage of the C. elegans worm from single cell to the adult hermaphrodite, which has 954 cells in it, not counting germ cells, is known. It's like a family tree. So you can look at an egg, you can take the dead eggs and look at them, see how many cells are in them, and you will know at exactly which cell division this lethality halts development, and therefore you would know a better idea about what that gene is actually doing in development. And having it held as a balancer like this would mean, well, this would be tough, because you wouldn't want to do this, because the dead eggs from UNC75 would fool you with the dead eggs from the lethal, so you would want something else, right? Like one that has a green fluorescent protein in it. Yeah, and so you would pick single worms that are green to a new plate, and anybody who had only green progeny would throw those away. But they would only be 1/4 of the total produced. Alright, so enough of that, because I don't want to get you too far down. But it's really cool stuff. So the reason I'm putting this in here is to try to give you guys an idea that all this stuff we're learning here, like who cares about inversions and translocations? I do, because I use them every day in my research for purposes of maintaining lethal and sterile alleles. And understanding how they work and why they suppress recombination is important. Anything else for Chapter 8? You're being uncharacteristically quiet. No. Okay, so any more questions for Chapter 8? Yeah. Nope, because we didn't do that in class yet. That's why. So we didn't get to that yet. So let me just give you a. So we didn't even do this yet, but let me just show you. I said I would talk about Robertsonian translocations coming up, right? So here's what it is. 2 chromosomes where the centromere is really close to the end, so either acrocentric or telocentric, and you get a break here and a break here, and you want 2 chromosomes get made into one bigger chromosome, and then the little piece down here is usually small enough that it gets lost or whatever. So, and then this is fine because it gets the centromere from one of them and it keeps going. And I'll show you, we'll talk about it tomorrow, but you have one of these. So look, humans have human chromosome 2 is actually a Robertsonian translocation that happens sometime after humans and all these apes separated as lineages. So chimps which have a genome that is 99% the same as ours don't have this. And also gorillas and orangutans, so they all have 2 different chromosomes. But all the genes on this chromosome match up with the left side of human chromosome 2. And all these genes on the other chromosome match up with the right side of human chromosome 2. So sometime after, the common ancestor that here's the lineage that humans came from, and here's the lineage that all the great apes came from, split off, these two chromosomes broke somewhere in here and came together to make a single chromosome. So, but we haven't talked about. We'll talk about it again tomorrow. So, and you could tell. So we're 2n equals 46. They're 2n equals 48 because they got 2 extra chromosomes. This, our 1 chromosome used to be 2. So you can see. This is an actual event that happened. All right. So you had a question back in Chapter 5, right? And what was it? I don't remember. Oh, was it sex influenced and sex limited? Okay. Yeah. Sex limited and sex influenced. Let me... What? Oh, sex linked versus sex limited. Let me talk about sex... What? Okay. So, sex influenced or sex limited versus sex... I'm almost done, and then I can come pull the pipette. Sex influenced or sex limited versus sex linked. Sex influenced and sex limited are on autosomes. Sex influenced and sex limited are not on the X or Y chromosome. Sex linked is, and usually when we say sex linked, we mean X linked, because besides SRY, there's nothing we've talked about that's on the Y chromosome. So, from the last exam, there was a question about how do you tell the difference between something that's on the X chromosome and something that's autosomal, right? And the way to do that is to do reciprocal crosses. All right, give me a second. I'm going to have to download this again. Yep. Okay, so how do you tell if something is sex-linked versus autosomal? You do reciprocal crosses. Reciprocal crosses mean in one case you're crossing a true breeding mutant with a true breeding wild type version. But in one case, you're using the males as the wild type, or the females as the wild type, and the males as the mutant. And in the other case, the females the mutant, and the males the wild type. See how that works? Okay. Yep. So, if you want to figure out whether something is sex influenced or not, you will just do a regular old cross, right? In this case, you have two heterozygotes and you cross them to each other. You can tell something is sex influenced if the males and the females have different phenotype ratios. At the very basic level, sex influence just means a trait or characteristic is dominant in one sex and recessive in the other one. And it's due to the different hormones, right? More testosterone in the male means that you only have to have one B allele to be bearded, whereas in the female you need two. So if you have a cross of two heterozygotes like this, you're going to get a 1 to 2 to 1 genotype ratio, same as it always is. But if you're in the males, both the BB homozygote and the BBB plus heterozygote are going to be bearded, and only the B plus are going to have no beard. So in males, beard is dominant. And in females, it's the opposite. You have to have two BBs to have the beard. So in both cases, you get a 3 to 1 phenotype ratio. But which one is the 3 and which one is the 1 is opposite for males and females. So, if you look at this right here, see that? It's a 1 to 2 to 1 genotype ratio. Here is a 1 to 2 to 1 genotype ratio. You don't need to make two different Punnett squares. It's the same Punnett square with the same 1 to 2 to 1. Just explain what would happen, right? I mean, if you want to make two Punnett squares, both of them would have the exact same genotypes in every square. It's just the three squares you circle for having a beard in male would the two in the middle and the other one would, only one of the three would have it for the female. So the genotype ratios are the same, but the phenotype ratios are different, and what's dominant and what's recessive depends on whether you're a male or a female. Sex limited is the same thing, except that no matter what you get, genotypically, one sex can't show it. So here, no matter what phenotype, or no matter what genotype you get, even if it's the little h, you don't get the rooster tail here. You get the little dainty little hen's tail or whatever it is, right? Does that, are we okay? These are not, you can tell that these are not sex linked because, here, let's go back. You can tell these are not sex linked because look, you have a male and a female here who are the exact same genotype. They're both heterozygotes. They're both heterozygotes, and yet look, one has a beard and one doesn't. Right there you could tell that it's... It would be better to just do one Punnett square and then below it just say, do one Punnett square and just do 3 to 1. You could just say, for males, BB blank is a beard. But for females, you have to be BB homozygote to have a beard. BB B plus or B plus would both be beardless for the female. If you did 2, you would not be wrong, but both Punnett squares would need to be identical. You could do 2 Punnett squares, and you could circle the 3 that's got a beard for the male and 1 and put an arrow to the one that doesn't. And in the female, you could circle the 3 that don't have a beard and put an arrow in like this one does. The most important thing is for you to be able to explain what is going on, and that is having the same genotype gives you a different phenotype depending on whether you are a male or a female due to differences in physiology between the two sexes. Yeah. That's the reason why. The phenotype of the offspring is determined by the genotype of the female parent. So this one is sinistral because its mother was little s. This one is dextral because its mother was little s blank, or s plus blank. This one, these are all sinistral, even though they have an S plus. Because of this, the genotype of the mother determines the phenotype of the offspring. And then in the F2, the genotype of the mother, S plus S, determines the phenotype of the offspring. These are all dextral, even though she is sinistral. She is sinistral because her mother was SS. Her progeny are all dextral because her genotype is S plus S. It's the genotype of the mother. So the way that everybody gets confused on this usually is that you're going to say the phenotype of the offspring is determined by the phenotype of the mother, but that's not true. It's the genotype of the mother. And the reason for that is that it's whatever, usually it's the either proteins or RNAs or other goodies that the mother loads into the egg that get this program started before you get gene expression from the zygotic genome itself. Good, good? Okay, that was yours, that was yours. Anything else, or are we all done? Everybody who's here now signed this, right? You signed this?